Every cell in your body carries DNA, a long piece of information or sequence of information that is returned in just 4 letters. But DNA itself doesn't perform any function. So instead, it acts like a blueprint. If it has to build something useful, like a protein, for example, the cell should first read it and interrupt this information. So that is where the genetic code comes in. It's a system that allows the cells to translate DNA instructions into proteins, the molecules that actually carry out life's processes. In this video, we'll explore how this code works and how a simple sequence of nucleotides becomes a functional protein. So let's dive in. Now let's understand that how the information flows inside a cell. Genes are specific regions of DNA that contain instructions for building proteins. First, the information that has to be first copied into a molecule that can carry that information and later on build proteins, right? So that is done by a molecule called the messenger RNA or mRNA. So as the name suggests, it's a messenger. It carries the information from the DNA to the ribosome, okay, to the place where proteins are, made. So the process where the information is copied into the molecule called as the messenger RNA or mRNA. This process is called as transcription, okay. So basically the DNA transcribes the information DNA is transcribed into messenger RNA. All right. Now, next thing is that the mRNA moves to the ribosome. So this is why mRNA is used primarily. Okay, the main role again, right? Since it can move, it can act like a transporter. It can move. Then it goes to the ribosome. So ribosome is a site where protein synthesis occur, and it is read and then so the instructions are read and then you build a protein. So this step is called as translation, right? Where the ribosomes read mRNA, right? So during the translation, the genetic code, the cells use the genetic code to basically determine which amino acids are added and in what order. So from the gene to protein, every step actually depends on correctly interpreting this code. Okay. Now let's discuss what exactly is genetic code, right? So a genetic code is a set of rules that tells the cells how to read the sequence of bases in mRNA. So these bases are read in the groups of 3 called as codons. So each codon corresponds to a specific amino acid. For example, as you see in this figure, AUG together, it is a one codon and it signals the start of protein synthesis and it codes for amino acid methionine. So as the ribosomes start to read one codon after another, the amino acids are added in sequence, so it forms a protein. In this way, the genetic code converts a simple nucleotide sequence, which is made of just four nucleotides into a functional protein, a functional molecule, right? Now let's discuss why the codons are triplets. So why are they 3 bases per codon? Why not just 1 or 2? The genetic code is built from 4 nucleotides, that is A, U, G, and C, which is adenine, uracil, guanine, and cytosine. If the amino acid were coded by just one base, we would just have 4 possible combinations, and that is not enough. Even if you have 2 bases, we'll only get 16 combinations, which is still insufficient to represent all the amino acids. But if the cell uses 3 bases at a time, it creates 64 possible combinations, 64 possible codons. And that is enough to encode all the 20 amino acids plus the start and stop signal. So that is why the codons are triplets. So the triplet nature of codons, they help to ensure the completeness of genetic code and also the flexibility. Now let's understand codons and let's see how they are organized. The genetic code is made up of, or it consists of 64 codons in total. Out of these 64, 61 codons, they specify amino acids, right? And the remaining 3, that is UAA, UAG, and UGA, all the black highlighted ones, they are they signal for stop codons or stop signal, which means they signal the end of protein synthesis. Each codon corresponds to only one amino acid, so it makes the code very precise and reliable. So scientists represent this information using a codon table. So codon table will help you to quickly identify which amino acid is encoded by a specific codon, right? Now let's discuss understand the key properties of the genetic code and why it is very reliable and an efficient system. So the first property is that the code is degenerate, which means that the multiple codons can specify the same amino acid. So for example, guanine and GAA and GAG both are separate codons, but they code for the same amino acid, glutamic acid. So this provides flexibility and it reduces the impact of mutations. Second is the code is unambiguous, which means that each codon specifies only one amino acid. For instance, AUG always codes for methionine and nothing else. Third, the code is non-overlapping. So codons are read one after the other in sequence without sharing bases. So a sequence like this, like AUG, GUC, AA, these three together, read has three separate codons, not as overlapping ones. Finally, the genetic code is nearly universal, which means that same codons code for the same amino acids in almost all organisms, from bacteria to humans. These properties ensure that the protein synthesis is not just accurate, but also consistent across all walks of life. Now let's understand how the code works and let's see how actually translation begins. Protein synthesis begins at a specific starting point. The starting point is the codon AUG, which acts like the start signal. So it not just codes for amino acid methionine, but it also tells the ribosome where to begin reading the mRNA. This is important because it establishes the reading frame, which is the correct grouping of codons. Once the ribosome identifies AUG, it starts to add amino acids in the correct sequence. If the reading frame is shifted even by one base, the entire protein can change. So the start signal, start codon, ensures that the translation happens at the right place and in the correct order. Once this process starts correctly, it should also end at the right point. So just as translation needs a clear start, it also needs a clear ending, proper ending, right? So some codons they do not code for any amino acid like UAA, UAG, and UGA. Okay, they act as the stop codons. So when the ribosome reaches one of these codons, it signals the end of translation. At this point, special proteins called the release factors, they go and bind to the ribosome. So they release the completed protein from the protein chain from the ribosome. So this ensures that the proteins that are made, they are having the correct length and structure. All right. Now let's move on to see. Now we have seen how the genetic code is written and read, but how is it actually translated into a protein? Right? So that is done by another RNA, that is the transfer RNA or tRNA. So transfer RNA or tRNA, it acts like an adapter, or it acts each of these tRNA, they help in carrying out, they have a specific amino acid to the, it helps carry a specific amino acid to the ribosome. Okay. And it also has an anticodon, right? Anticodon, which is basically the complementary sequence to the codon on the mRNA. So when the anticodon pairs with the codon, the correct amino acid is positioned in the place. This ensures that the protein is built accurately, one amino acid at a time. The ribosome then links these amino acids together. It forms a growing polypeptide chain. In this way, the tRNA acts as a molecular translator, converting genetic information into a functional protein. Fine. So now let's discuss an important question. Okay, an interesting question that now how can the limited number of tRNA molecules recognize so many codons? That is explained by the hypothesis called the Wobble Hypothesis, which was proposed by Francis Crick. The key idea is that the pairing between codon and anticodon is not equally strict at all positions. While the first two bases pair precisely, the third base allows some kind of flexibility or wobbling effect. Because of this, a single tRNA can recognize multiple codons that differ in their third base. This explains why the genetic code is degenerate. It also makes translation more efficient because there are fewer tRNAs needed. At the same time, Wobble reduces the impact of small mutations, especially at the third position. So this gives not just flexibility, but it adds efficiency and stability to the genetic code. Now let's bring everything together, right? The genetic code is a set of rules that tells how information in DNA is converted into proteins. The molecules that basically control structure, function, and all the activity in every cell. Even a small change happening in DNA can alter a codon. This can lead to different amino acids being added, which can change the structure of the protein. And when the protein structure changes, its function also changes, so it also will have significant biological effects. So that is why the genetic code is so important, because it connects the information that is stored in DNA to the physical traits we observe in living organisms. So from the cellular functions to genetic diseases and even modern biotechnology, everything depends on how this code is read and interpreted. Finally, a sequence of just 4 letters organized into precise triplets is all that takes to build complexity of life. And the genetic code is the key that makes this information transformation possible. So thank you everybody for joining me. So I hope you liked the video, right? Please like it, share it, and subscribe. More will be coming. Thank you.